Hi everyone. Today I will be discussing one of the most challenging questions in the modern medicine. Why is the developing a vaccine to the HIV-1 virus so difficult? This question has puzzled scientists for decades. As well we will see, the answer lies in the unique characteristic of the virus. So let me start by providing some background on HIV-1 infection. When the HIV-1 virus enters the body, it has very specific target, CD4 plus T cells, which are crucial type of immune cells. The virus binds to these cells, fuse with their membrane and release its RNA. This RNA is then reverse transcribed into DNA and integrated into the host genome. This integration allows HIV to replicate and produce new virus particles, gradually weakening the immune system. Over time, if left untreated, this leads to AIDS, where the body becomes vulnerable to opportunistic infection. Now here's the crucial point. Epidemiologists around the world believe that inventing an effective vaccine is the most viable option to control the HIV-1 pandemic globally. However, vaccine development efforts face several challenges due to the unique features of the HIV-1 virus. So how most viral vaccines work? To understand the challenges, let's first look at how most viral vaccines work. Generally, most vaccines mimic the natural infection and contribute to producing neutralizing antibody response. These neutralizing antibodies are produced by long-lived plasma cells and immediately inactivate virus particles when they encounter them. Additionally, vaccine-induced memory B cells that contribute to secreting neutralizing antibody upon future exposure. Importantly, the effectiveness of vaccine is assessed by their capability of producing adequate neutralizing antibody level that providing protection against infection. This brings us the heart of our problem with HIV-1. So what's make the HIV-1 difference? The HIV-1 virus has broadly diversified HIV variants circulating worldwide. Therefore, the neutralizing antibodies produced by vaccine must be capable of effectively neutralizing all these HIV variants to provide comprehensive protection. To achieve this target, the selected immunogen should be produce a broad range of neutralizing antibodies that can neutralize all variants. Unfortunately, it is extremely difficult to identify the ideal immunogen to produce such a vaccine. As a result, this virus is often only partially neutralized. This mechanism drives the generation of new resistant viral strain, making the problem even more complex. So how can we resolve this issue? Generally, most vaccines target both humoral and cellular immune system to train immune cells against infection and producing neutralizing antibodies. However, as I mentioned above, attempting to produce broadly neutralizing antibody against the HIV-1 virus may be less effective using traditional approach. Therefore, scientists are trying to different approach. They are attempting to induce cellular immune response throughout the vaccination against HIV-1 virus infection, focusing on different arms of the immune system. So this brings us to T-cell vaccines. These T-cell vaccines specifically target both CD4 plus and CD8 T cells and train these cells against infection. Now remember that CD4 plus T cells are the primary target of the HIV-1 virus. Scientists believe that training CD4 plus T cells against the infection may be helpful in reducing damage to CD4 plus T cells when actual infection occurs. On the other hand, training CD8 T cells may be beneficial in increasing the capacity to identify the infected immune cells during the infection and remove these cells from the body throughout the cytotoxic response. Let me now walk you through some key clinical trials that tested this approach. These early attempts span from 2003 to 2009 and show us the evolution of HIV vaccine approach. The first trial in 2003 used recombinant monomeric GP120 vector as an antibody vaccine. Unfortunately, this trial showed no efficacy in preventing HIV infection. The second trial in 2007 employed adenovirus vector encoding HIV internal protein GAG as a T-cell vaccine. Again, this trial also show us no efficacy. However, the third trial in 2009 utilized the canary pox virus vector encoding HIV internal protein GAG as both antibody and T-cell vaccine. Significantly, this trial showed the limited efficacy, representing the first glimmer hope in HIV vaccine development. In conclusion, the development of an effective HIV-1 vaccine remains challenging due to the virus' unique characteristic, the broad diversity of HIV-1 variant, and the difficulty in generating broadly neutralizing antibodies continue to pose significant obstacles. However, innovative approaches including T-cell vaccine and new delivery technologies like mRNA platform offer promising avenue for the future research. While progress has been slower than hoped, each clinical trial provides valuable scientific insight that inform the new generation of the vaccine candidate. Thank you for watching. Please subscribe for more videos. Thank you.